Good morning and good afternoon to all of you for joining our live webinar today, Secondary Transport Inhibition and Regulation by SSM-electrophysiology. Today's webinar marks the second of 5 upcoming talks that will make up the part of our 2021 Research Insights Transporter webinar series. My name is Jason Villagomez, Marketing Manager of Nanion Technologies, and will be your moderator for today's event. But before we get started, my colleague, Dr. Maria Bartmus, Senior Scientist and Product Manager of the SURFR team, would like to say a few words.
Hello, everybody, and warm welcome also from my side and on behalf of our whole team, including Andre and Cecilia, who some of you might know. So as Jason mentioned, this is now the second of 5 webinar sessions we're hosting this year. So welcome back, everybody, if you already joined the first session. Usually we host only 1 or 2 such webinar every year, but with so many conferences and meetings being canceled this year, we decided to go for a whole series. And we are very pleased and thankful that so many scientists accepted our invitation to speak about their research projects here. Today, special thank you to Camilo and Christine. Thank you for making this possible. You can find all details about the next sessions on our webpage, and we will post also a link in the event chat shortly. Something we are also very happy about is the big number of participants so far. So this really makes worth the whole effort. So also thank you for participating. We hope you enjoy the session, and I really encourage you to make this an interactive event by asking all the questions that might come up during the talks. We have planned a dedicated time slot for this. Jason will let you know more in a second. So if you're interested to try SSM-based electrophysiology yourself, or maybe you become so during the talks, you still have the opportunity to apply for our Instrumentation Grant. This includes an instrument, all consumables, and also support, scientific support for half a year. So you can apply with your project until July 15th. So we will also put a link in the chat in a second. Another option to try this technology is, in particular, if you're interested in HTS format, is to get in touch with AssayWorks. That is a CRO providing assay development and screening services using the 96-well server. With that, I hand back to Jason, who will guide you through this webinar. I'm looking forward to the talks, and I hope you enjoy it.
Thank you, Maria, for the exciting updates to look forward to. Joining us as presenters today, Professor Dr. Christine Ziegler, University of Regensburg, Faculty of Biology and Preclinics, Institute of Biophysics and Physical Biochemistry, Structural Biology, Biophysics, and Professor Dr. Camilo Perez, University of Basel, Biocentrum, Center for Molecular Life Sciences. Today's presentation is as follows. Christine's presentation is entitled The Potassium Switch in BetP, From Coupling to Regulation. Camilo will then proceed with his presentation entitled Selection of Transporter Targeted Inhibitory Nanobodies by SSM-based Electrophysiology. A little bit of due diligence. Upon completion of the presentation, we will hold a live Q&A session with the presenters, and as such, welcome you to submit questions at any time throughout the course of the presentation. You can log a question in the chat window found on the right hand of your screen via the menu bar. Your message will come directly to me and will be added to the queue. If you happen to experience any technical difficulties at any time, please inform us via the chat box. Additionally, today's presentation will be available on demand shortly upon completion of the event, inclusive of a transcribed copy of the Q&A session. I will now turn over the presentation to Christine for the start.
Yeah, thank you, Jason, for the introduction. I hope everybody can hear me and see my slides. Good. So Yeah, today I would like to talk to you about transport regulation and in fact how this regulation is now controlled by lipids and internal potassium in the sodium beta importer BetP. But before I start, I really would like to acknowledge first the people who did that work, Veronika, Gregor, Katharina, and Christian. So in my group, the question of how lipids can in fact mediate transport regulation is really a big issue. And it is not only a specific interaction of lipids, but really the interaction and a signal transduction over the whole membrane as a bulk, because the membrane as an entity is kind of interconnected and can very well transduce signals, for instance, by this model of elastic response, where you have the interaction between lipids like little springs. So it's a very physical approach to think about a signal transduction. So to understand that, you need structure, you need to see the lipids, but you also need functions, essentially to have function and dynamics based on spectroscopy. And the way now structural biologists approach that is by combining different methods, and we call that an integrative structural biology approach. But aside of the methods, you need a good target to investigate such lipid-mediated regulation. And such a target is, in fact, the sodium beta importer BetP. It's an osmolyte transporter involved in osmoregulation. And that is especially the case when under hyperosmotic conditions, the cell requires osmolytes like betaine, and this has to be accumulated really against the concentration gradient. So BetP is such a target, not only because it's regulated, but because it has such a nice correlation to the lipid species in the membrane. And you see that very nicely when you look to the betaine uptake rate and how this rate is now changing with osmotic stress. And you see that you need a certain threshold before that transporter, which is silenced, then really go into full activation mode. And for BetP, this depends really strongly on how much negatively charged lipid we have here. Now, important is to realize that the upregulation is important, but much more important is the downregulation, because no cell wants to have a lot of osmolytes pumped into it without any stress. So activation, downregulation, big issues in BetP. Interestingly, in BetP is not only the interaction with lipids, but the requirement to have a certain amount of internal potassium to come to a full activation. In fact, this internal potassium can be used as a kind of pre-activation, and you can mimic the same betaine uptake rate osmo profile also by just increasing the internal potassium concentration. And you see here that you have kind of binding constant, which is around 220 millimolar. So we can say, well, that's the normal condition in the cell. And that's true. This potassium stimulus is not really a physiological one, but it is very practical when you want to do biophysical investigations. So what BetP requires is to somehow monitor the hyperosmotic stress, and that is performed by the so-called osmosensor, which is a C-terminal domain, which is folded by itself. And now the intriguing question here is, how is now the osmotic stress signal transduced? And that is already known that might be a change in the bulk membrane, as I have introduced to you. So to answer now this question, how regulation occurs, the first step is always the mechanism, which would mean how structural elements are now affected by this bulk membrane interaction. And very nicely, BetP, also being a member of a quite specific family, the BCCT family, luckily exhibit the famous Lloyd T fold, which makes mechanistic considerations much more easier. So important for today is to look at the substrate binding sites and the coupling mechanism, which means we have to look at the way the two sodium ions which are required for transport are now really related to the binding of the osmolyte. And the nice thing in Leutifort, it has this 5 transmembrane inverted repeat motif, is that it's super symmetric. So if we now look here into the binding pocket, we see the 2 sodium ions. Here is the Na2 side, and here is the Na1 side. And here in the middle, we see the osmolyte beta in this tryptophan box, nicely nested with its 3-methylammonium group. And we can anticipate that also the same structural elements are involved in binding. There are different residues now involved. So aside of the Leutifort transporter core, BetP has additional elements like the osmosis sensor, which I mentioned already, but also here at the periplasmic side, we find an amphiphatic helix, which we call H7. And this helix is getting really important in the trimeric state of the protein, which is necessary for the downregulation and activation. And we see here that along this H7, we have major trimerization contacts. If we look now here to the side view, we can see 2 things. We see that we have an hydrophobic cavity where lipid densities are found. And we also see the osmosis sensor here. But also this is a trimer, we see that it is kind of asymmetric. So at least it is asymmetric resolved. And important is now as a take-home message that this trimeric state is really important to provide a platform not only for protein-lipid interactions, but also for lipid-lipid interactions, especially when it comes to the downregulated state. And this downregulated state we were recently able to solve in a cryo-EM structure. And you see here already that different to the X-ray structure in DDM, we have now our osmosis sensor nicely resolved as a highly symmetric structure. And the C-terminal domains are also oriented differently. They range to different parts of the protein. And for sure, also in the cryo-EM structure, we have lipid densities. If we look here a little bit closer, we see that we have a lipid density in the cavity, but not as much as the X-ray structure. And we have a cardiolipid, which kind of fix now the osmosis sensor against this loop of the adjacent protomer. And there are a couple of consequences from this downregulated state, which I will not go into today, just to tell you we have changes in the binding sites, and we have changes here, interactions with of the C-terminal domain with what we call the coupling scaffold. So for sure now the question is, if we want to look at regulation, what will change? For that, we have now determined a cryo-EM structure in the presence of our non-physiological stimulus, potassium. And in comparison now with the X-ray structure, which we assume represent a pre-activated state already, you can now very easily see what happens. So if we start here in our very symmetric, fully downregulated state, and we now come with potassium, we increase potassium, we see that even in the cryo-EM structure, we have a break in symmetry. The C-terminal domains kind of start unfolding, and you see that they're doing that differently. Every protomer is doing a different kind of degree of unfolding with consequences to the lipid interaction. And if you look now here in the pre-activated X-ray structure, it seems that like these lipids are sucked into that cavity. And something which is also very nicely to monitor is that the cardiolipin you see here, which in the downregulated state fix the C-terminal domain, is moving like with the C-terminal domain downwards. And you can look at that vice versa, which would tell you if something comes from the bulk lipid, the lipid which is attached there can easily now control and move the C-terminal away. So what is the consequence now of this sucking in of the lipids to BetP? Well, the first thing we see that is the whole trimer architecture change. So if you overlay the cryo-EM structure and the X-ray structures, you don't have the same way how this trimer is oriented with respect to the membrane. And you see this in a very simplified scheme that in the downregulated state, the kind of this cavity is narrower, and the different protomers seems to arrange more parallel to the membrane normal, while in the activated X-ray structures, they kind of slightly tilt. And even the C-terminal domains detach from the membrane part and can, after unfolding, switch back. And for sure, the consequence I told you already is that lipids can move in and stabilize that, or the other way around, that the lipids move in first and stabilize this. So here we have the key to the membrane stimulus we were looking for quite a long time. But now the question what we have today is, how actually this kind of conformational change result in a difference in activation, which means for BetP that in this situation here, the Vmax suddenly increases really a lot, 5-fold, 10-fold. Yeah. So where is now the angle that this activation takes place? And already quite some time ago, the group of Reinhard Kremer, who is kind of the father of BetP, found out that mutations in the osmosensor, especially here in the knee or here in the middle, where the C-terminal kinks, or here at the end where it interacts with the other protomer, affect to some degree, to some extent, the affinity to sodium. So there seemed to be a link to the sodium coupling. And we also saw that to some extent by doing fluorescence measurements on a BetP, where we used the beta in KD to kind of titrate now sodium and determine a sodium KD at different potassium concentrations. And if you look at this here roughly, you will see that the KD for sodium is increasing. Yeah? So the affinity is decreasing, and we have a kind of strange phase here, a biphasic behavior when we do this transition from downregulation to upregulation. And clearly this asks for functional investigations. So let me summarize that here, that you understand the problem. We have not only a catalytic cycle, which we want to understand, we have a regulatory cycle, and we would really now like to see what happens here on this catalytic cycle if we activate. And if we now look here at these two states, this outward apostate and the outward substrate binding state, it is important now to see what happens now, especially during sodium coupling, because that is our working hypothesis that somehow during regulation, this is affected by, the effect is at the sodium binding sites. So Lucy Forrest did a couple of really nice simulations, which I want to show here again. And if we look here at the sodium 2 site, sodium 2 binding site in this outward facing state, we see a very nice coordination in this conserved state, but it's not really stable. Yeah, so you will see that the residues are moving slightly, and then that water will rush in. And this is even worse in the Na1 side in the outward facing state. So in BetP, to have a really stable coordination of sodium beta-ene is required. And you see that clearly here in the Na2 side, where this methionine, which coordinates the beta-ene, is kind of then forcing here the site to get in a stable state. And in the Na1 side, interestingly, this cation pi interaction is kind of disrupted by beta-ene binding and moving the sodium ions here closer. Yeah. So to now try to grasp what happens when you want to now mix the catalytic cycle with the regulatory cycle, the best thing to do is mutagenesis. To understand now, to have functional different mutants, which you then can investigate with different methods. And in our case, we looked at the BCCT family, where BetP belongs to, and found a class of transporters which had in the sodium 1 side, instead of this threonine here, an isoleucine. And one of the species is described to be potassium coupled. And we solved the structure here of this mutant, and you see very nicely that this site, by this mutation, kind of transforms now in an always occupied Na1 site. And the mutant we created was indeed able to couple to potassium, also with a very reduced Vmax. So what we did then was to compare the wild-type BetP and this mutant using now a time-resolved methods. And we started here with stop flow fluorescence measurements, which is a perfect tool if you really want now to go into rate constants and into the kinetics. And what we look here at BetP is in fact a two-step binding model, where we first have the binding of our substrates and then the transformation or isomerization to this closed state. And that is really where this fluorescence, protein fluorescence, can be extremely helpful. So if you measure this now, the important point is that you would like to have a rate constant, but what you measure is a fluorescence signal. So what you have to do now is to do a fit, and this was done either as a single exponential or a double exponential, which gave you then these K observed, and from that you can now deduce the rates for forward reaction or backward reverse reaction, and can also deduce the KD here from the rate constants of this first step. So now an important point is to remember that also this looks here very nice. You always have a apparent rate constant because you're looking at the membrane transporter and not at an enzyme. It's not a product you at the end create. So let us just have a look how in stop flow here BetP and the mutant change. So we tried different conditions like just sodium or sodium and potassium alone. And the wild type, you will not see any binding with potassium alone. But if you just now look at these apparent KD values, you see that neither for BetP wild type nor for the mutant, you have so big changes, which tells you that here, in fact, the first sodium site is important, the Na2 site is important, as we deleted the Na1 site. It gets now much more interesting if you look at the following rate constant forward and reverse. And there you see that BetP, the wild type, is quite fast forward, yeah. And if you then add potassium, it seems to slow down. If you compare that now with the mutant, you see it is a little bit different, but not so significant. It's more interesting if you now look at the reverse reaction, where you see that this close to second state is kind of depopulated much faster here with KCl when potassium is around. However, in the mutant, where the sodium one site is constitutively active there, you don't have this, this backward reaction. So it's kind of stuck here in this closed state. Interestingly, the situation is different when we have only potassium, which is in good agreement that we have a potassium transporter. But one thing which is really interesting is when you now use both sodium and potassium. There you come to the scenario that a single exponential is not sufficient and you have to go to double exponential. And then you will discover that one of your components is really, really fast. And to look now more in detail into this interesting behavior of the reverse time constant, we thought it would now be an important point to do that, not in DDM, not in detergent, to do that in proteoliposomes and now use SSM. So I will not go into the technique of SSM, probably you know that much better than me. I just show you the buffer conditions we had for our non-activating buffer and our activating buffer. And important is that in all the measurements I show you today, we used the sodium as a driving force. So we used this ionized sugar and then jumped to sodium, and we kept the substrate concentration constant in the individual points. So what you get are these transients, and now you can do different things with it. As you already know, easiest would be just to look at the peak current potential, which gives you an information as a current, which would mean Coulomb per second. So it is already kind of in a rate region. You can also look how this peak position shifts, and you can do the integral. But we have appreciated a lot the information you in fact get from this slope of the decay time to understand more complex transport pattern. So let's just have a first look for the wild type, for the peak current and for the integral, which you see here plotted in red is the peak current, and in blue we have the translocated charge. So the question is, how can we analyze that? And we decided to do just a Hill equation fit, which gives us a constant, which we have called K half. And that would means we have one for our peak current potential, and we have one K half for our translocated charge. So they look not the same. Also, the Hill coefficient is pretty much the same value. So what we are looking at here. So in the case of the translocation charge, we know this is a kind of endpoint measurement. And what we have here is a thermodynamic equilibrium constant, which kind of gives you the free energy difference between these two states in the two steps I have described. So, and if we go now here to the peak current potential, and I mentioned is already something like 1 per second, it is like a reaction rate like constant, but at that point it's hard to tell what it is. It is kind of a mixture. It's not like the stop flow, a pre-steady state is a mixture of these states. And we will come to that point a little bit later. So let's have a look to the mutant, and immediately from the transients, you see they look really different. And if we then look at the peak current potential, and again at a translocated charge, we see here that the peak current potential looks a little bit like Michaelis-Menten, but we have a different Hill factor already for the peak current. Question is, how significant is that? And if we compare that here to the wild type, our rate constant is quite reduced. If we also look now here at the data points, we see that after 5 millimolar, you have you see here that there's a decrease. And the question is that real, where that comes from? And more interesting is if you look at the integral. This is not a charge which is kind of accumulated, it's decreased. Can that be? So these effects require now, as I said, the famous decay time, which really helps in that case to look what happens. So for the wild type, it was a quite straightforward way. You take a single exponential, and again, instead of the decay time itself, we just take the inverse decay time, which provides us the again a rate constant, which we can plot. In the case of the mutant, this was not so easy, yeah, and we tried like a single exponential with a linear term, double exponential, with which then leaves you with 2 rate constants. So just to give you a hint how to decide here, you can always just try and see what fits better, but you could also just go and do the first derivative. And if you do the first derivative, you will see that here you have a quite different behavior of the wild type and the mutant. So in green is the wild type, and you see that that goes down here nicely and then kind of starts moving here towards the 0, while in the mutant you have here like again a minimum, which already gives you a hint that there is something more complex going on. But important, with all these fitting methods, at least is what we saw, is that it really depends also on the substrate concentration. And a take-home message is that whenever you would like to fit decay times, it might not be possible in the same way for all substrate concentrations. So here for the lowest concentration, you do not see that behavior. But again, that's a good tool to investigate what's happening. So let's just see how our rate here looks when we plot it again against the substrate concentration, and we see is a very nice plot which finally gives you with the rate, provide you the rate constants you want, because you plot a rate which allows you now to use, in fact, the same way to fit now the 2-step binding process. And what you see here are now different things. The Hill factor is pretty much what we had before. We have a real KD now, and we have a forward and reverse, which differ just by a factor 2. So now, if we go now here to the mutant, the situation is completely different. First, we have 2 rate constants. We fitted double exponential, and you see that they behave really strangely. First of all, we have a very fast component where we touch here the resolution limit of the sensor. In that case, we use the 3 millimeter sensor, which tells us that here, in fact, after like 1 millimolar, the data cannot analyze with that, yeah, and cannot be considered in our fits. Then we have a slower part of the mutant, where we got data, and that we see here, which are just not reasonable for the lower part. Yeah. So using now the decay time and plotting this inverse decay time really helps you to judge if something, if you have a more complex process which might not be consistent. This you can also see by just plotting here the peak current time, which means where you find your peak current. And in the case of wild type BetP, you see that this is very nice in the range before we are in the region where we have the void time. So when we are at the dead time of our sensor, while in our fast mutant, we see immediately that we already around this between 1 and 2 millimolar, we are below that. And that is exactly the explanation that with this mutant, we cannot just use the same experimental conditions like the wild type, because we are here in the limit of the machine, which we could see very nicely by plotting the decay time. So now the big question is, can we see regulation? Can we see the potassium switch in SSM now, knowing now the way to analyze that? So first we start with the peak current potential, and I remind you, this is kind of our rate information, and very nicely we see here that we indeed have a difference between our zero potassium state in the wild type and our non-physiological stimulated BetP, activated BetP. And most interestingly is that we have a change in the Hill factor here, yeah, which can be clearly seen here in the beginning. Now we can look at the integrals, and again, the integral translocated charge makes a lot of sense. You see here that we reach the same endpoint, but the way this is now transformed, this is now achieved with a 300 millimolar potassium is much more different than without potassium. And in fact, if you look again at this integral data at zero potassium, you see a quite similar behavior to the mutant, which just tells you you are here out of, this is not really transport you see without potassium. And at the end, to look again at the rate constants, beautiful rate constants with 300 millimolar, and at zero, again, you see the cooperativity coming up here, but you also see that there is some process here which change, in fact, one of the rate constants in a way that we again very soon reach the limit. Yeah. So what we can deduce now from looking at these different parameters is that, in fact, the NA1 site, which we kind of took out in the BetP mutant, is important, yeah, and is important in a negative way, which means when it's always there, the mutant in fact behaves like the down-regulated protein. So with that, I come to my last slide, and I want just to summarize a little bit what, in our opinion, you can do with SSM when it comes to such a complex alternating access binding model. So for BetP, we feel quite confident that we see transport because we could see differences when BetP was down-regulated, and we are also confident that we see regulated transport. The other point is that it would now be nice if you can inhibit transport, but that is a topic which will be touched by Camilo later. For BetP, unfortunately, we don't have any inhibitor, which is really a pity. But one thing which we will like to push further is now to put really name tags on these apparent rate constants. Yeah, you saw that this is at the moment not so clear, but we will continue working on that. So what is important as take-home message, what we learned from our studies on BetP, you have to respect the resolution limits in SSM, which is the sensor resolution, the void time, and you have to think about that the buffer conditions, substrate concentration, how much protein you have, that is an important parameter to adjust. And finally, if you see that your SSM data are not consistent, yeah, or the quality, that most likely this has something to do here with the latter point, and that it's also not so easy between a protein and its mutant to compare these rate constant, because the moment one step is missing and you look at a mixture of constant, yeah, it might, you might not see the difference. But this is something for the future. And with that, I would thank again everybody who contributed to the project. And I will give over to Jason again.
Thank you, Christine, for walking us through a deeper dive into your work in BetP. I will now introduce Dr. Camilo Perez, who will walk us through selection of transporter-targeted inhibitor nanobodies by SSM-based electrophysiology. So with that, I hand over to you, Camilo.
Thanks, Jason. I hope you see my slides. Okay, thanks. Thanks for the invitation. I'm really happy to be able to participate in this lecture series, and I'm really happy to share today the stage with Christine, which was, by the way, my PhD supervisor. And today I'm gonna discuss about a project in the lab, which is about our secondary transporter, but specifically I'm gonna show how do we use solid supported membrane electrophysiology to screen transported targeted inhibitory nanobodies. So the work I'm going to present was done by a very talented PhD student in the lab, Natalie Berland, which is, by the way, still here. And she's with the people here looking at this talk. So in the lab, we investigate the different processes involved in the biosynthesis of cell wall in gram-positive bacteria. Just to give you an overview, the cell wall of gram-positives looks something like this. There are mainly three important components. One is a very thick peptidoglycan layer, which you can see here as the green blocks. You have, of course, the plasma membrane, and then you have two different types of biopolymers, which are ranked in the family of teichoic acids. But there are two of those. The first kind are these ones right here, which are anchored to the plasma membrane. They are called lipoteichoic acids. And the other ones are these ones right here, which are anchored to the peptidoglycan layer. In most gram-positive bacteria, the biosynthesis pathways of these teichoic acids are different, and they involve multiple different proteins. It has been shown that teichoic acids are actually very important for host colonization, addition, biofilm formation, immune evasion, and resistance to antibiotics. In my lab, we are interested in investigating membrane proteins involved in the biosynthesis of these teichoic acids. In particular, we are interested in understanding how is the mechanism of transporters that will translocate precursors that help to build up these, to synthesize these polymers. Another kind of protein that we are interested in are polymerases or transferases, which are also involved in this cell wall biosynthesis process. So transporters are extremely important, as I mentioned, because they are the ones that allow the translocation of water-soluble molecules across the membrane so that they can reach their final destination. Many of these transporters are actually specific for some pathogens, and this can become and has been actually very important for development of new antibiotics. And they are more accessible than cytoplasmic proteins, of course. And not only that, but okay, there are actually a bunch of these proteins which are actually very interesting from the point of view of their mechanisms, which are not only ranked among prokaryotes, but can go beyond or even to eukaryotes. Because they share similar folds and same families and so on. So the particular transporter that we are investigating in this project is involved in the biosynthesis pathway of a bacteria, which has actually a very interesting way to synthesize teichoic acids. Just let me put here my laser point. Okay, so this synthesis pathway is actually particular because, as I mentioned, there are two kinds of teichoic acids, right? But these bacteria synthesize both kinds in one single pathway. And the pathway involves two transporters. One is a choline importer, which you can see here, and another one is a flippase, which helps in the translocation of the teichoic acid across the plasma membrane. Today, we are gonna discuss the choline importer. So it turns out that actually these pathogens that have this pathway, they are not able to synthesize choline by themselves. So they need to uptake choline so that this choline is later attached to the teichoic acids. And then later on, when the teichoic acids are now sitting at the cell wall, these choline residues become the anchor points of a very important family of proteins, which are called choline binding proteins, which are depicted here as these mechanical arms. And these choline binding proteins, they come in multiple different flavors, and they help these pathogens to perform multiple different tasks like immune evasion, adherence, spread, and invasion of the host. So if you want to imagine this as something that shows up in a movie, we can imagine these as these robots that were shown in the movie The Matrix, where they have these multiple arms that have multiple different tools, and these help basically these robots to kill humans. So it's the same with this kind of pathogens. So we are very interested in understanding how this transporter allowed the transport of this vital molecule for this pathogen, and as it is essential for them to build up the choline binding proteins. So Natalie actually started her PhD with a high energy trying to crystallize this protein. The protein is very small, 32 kilodaltons. Cryo-EM by then was not yet reaching this size of molecules, especially membrane proteins. So we started doing hardcore crystallography, doing vapor diffusion, LCP crystallization. She was very successful having crystals of this protein, as you can see here, also in LCP. But unfortunately, even though we screened hundreds of these crystals, it was not possible to obtain diffraction of X-rays, which can go beyond 7 angstroms. It was only possible later on to obtain a data set at about 3.8 angstroms after we performed in situ annealing, which I'm not going to explain in this talk, and if somebody's interested, I can explain later. But in any case, even though we had a 3.8 angstroms data set, it was not possible really to solve a model that can show us side chains, because we only can see tubes basically because of the model inaccuracy. We use a model deposited in the PDB, so we have poor phases. That's when we actually became very interested in producing nanobodies against this protein so that it can help us to solve the structure. As you know, camelids and sharks have a special kind of antibody. So in contrast to conventional antibodies, we have a heavy chain and a light chain. These animals have only a heavy chain antibody. And the domain here, which contains the complementary determining regions that recognize epitopes in antigens, they can be actually isolated, they can be expressed. They are very small, highly stable, and they retain the antigen affinity for their antigen. So the people has been using this sort of tools for multiple purposes in biochemistry of proteins, including gadgets for structure determination, stabilization of conformations, inhibitors for catalytic activity, and there is even potential for having them as in applications like in the clinical. So these nanobodies can be produced using a classic method, which is what was used for many, many years, which consists, of course, of immunizing camelids, in this case, alpacas, isolation of B cells, cloning of the nanobody libraries, and selection, which was usually done by phage display. But there has been other methods which have been enlightened in the last years, which include an in vitro selection, which is based on construction of DNA libraries. And this is very good because there is no animal immunization, so we don't need to deal with the animals anymore. And in that case, the selection of these nanobodies is done by either phage display, ribosome display, or G display. There are multiple papers that discuss these different selection methods if you are interested in it. In this particular project, we collaborate with the group of Marcus Ziegler and his PhD, Cedric Hutter, which actually help us to produce synthetic nanobodies. So in this case, Marcus has a library of these nanobodies that we combine ribosome display and phage display to perform the selection. We were lucky to have actually multiple binders, so you usually make it 10, it depends of course on your protein, but we were able to obtain about 25 different synthetic nanobodies that bind to the protein. As you can see here, we see a clear shift in size exclusion chromatography profiles of our protein and then protein bound to the synthetic nanobodies. So at this point, we also wanted to determine how strong is the binding of these proteins, of these nanobodies to the transporter. So together with Marcus, we performed grating coupled interferometry to determine these KDs. And this is just a summary of the data that show us that we had KDs of around 40 nanomolar for all nanobodies. There were differences in association constants and dissociation constants, which you can visualize here from the decay, but overall they behave very similar. But at this point, so we were very interested in picking up or selecting inhibitory nanobodies because of the very important function of this protein. And well, they are just simply more interesting from the functional point of view. So we had in hands, in principle, two methods that we could apply to select these inhibitory nanobodies. One of them is the classical uptake assay, where we have a radio-labeled molecule, which in this case is tritiated choline, and we could have liposomes with or reconstituted protein. And then after the different times, we just measure accumulation of the radioactive choline at different time points. Another assay that we had in hands was overexpression or expression of this protein and in a special E. coli strain, which is devoid of choline transporters. And after some time, we just measure accumulation. This was done with the wild-type protein, which you can see here, and with some mutants. However, application of these methods for in a high throughput mode was already, let's say it was possible, but in the case of liposomes, this will be very laborious. This will require a lot of work and a lot of time and a lot of material as well. And in the case of the cells-based assay, we don't even know if this was possible because we don't know if nanobodies can cross the cell wall. So that's why when we came to our very interesting technique, solid supported membranes electrophysiology, which you all know, and we exploited the high throughput properties of this technique to screen this library of nanobodies. So I'm just going to briefly explain what actually what kind of experiments we did. So in a very first experiment, we do the classical protocol for SSM, where we have nanobodies on our monolayer. In an activating buffer, we have choline in our case, and then we just pass again non-activating solution to wash out the substrate. In the case of nanobodies, we have either inhibitory ones, we have them in both non-activating and activating buffer, and I will explain you in a minute why is this important. And in this case, we won't see, or we will see a decrease of the peak currents, as you can see here in the blue curve. This is, of course, a fictional data here. This is just to exemplify what we were expecting. And in the case of non-inhibitory nanobodies, we were expecting them to not bind and to not block transport, so that we were able to see a still transport of choline in our case. Now, as I mentioned to you, we have nanobodies in the non-activating solution and also in the activating solution, of course. Now, this is very important because, and we actually realized this after multiple experiments, where we, at the very beginning, we didn't have nanobodies in the non-activating solution. In that case, every time we performed this experiment, we did not observe inhibition at all. Now, this was something that actually we should have expected, because nanobodies and the binding of nanobodies to proteins follows a second order kinetics, meaning that the lower the concentration of the nanobodies that you use in the experiments, and we use 500 nanomolar, will also lower the rate at which this association process happens. So we realized that actually we had to have these nanobodies in the non-activating solution and have this pre-incubation, so that there is enough time for them to interact with the transporters in the protoliposomes. And later on, when we apply choline, we would see an effect. This is what was done. In our case, we tested multiple different times from 2 minutes up to 30 minutes. We saw that 2 minutes was optimal for all the nanobodies that we had in our set. And we the other thing that actually we did just to normalize all our measurements was to perform 2 measurements of peak currents, so non-activating, activating, non-activating, and discard the data or just leave it there. And only the 3 following experiments were used for data analysis. So we started with the most simple experiment, as I mentioned, which is a transport of choline in the absence of nanobody. Here we have the peak currents for choline titration. We don't have a significant noise signal when we have protein-free liposomes. And we performed these experiments because we wanted to see what are the saturation conditions that later on we will use for the screening of nanobodies. So in this case, we selected 5 millimolar choline. Now, with this 5 millimolar choline in all our experiments, then we follow to perform the screening with nanobodies. And we started with 500 nanomolar of nanobodies concentration. The idea of using 500 nanomolar was to be way above their KD constant, which, as I mentioned before, was about 40 nanomolar. So here I show you some examples. And first of all, we were happy to see that protein-free liposomes do not show also any significant noise current when we pass nanobodies or when we have nanobodies in our measurements. This is how you see here a peak current of protein-free without nanobody. The protoliposomes without nanobody. And here we see a non-inhibitory nanobody. Here, the green curve, there is just a mild decrease. And here, this is an example of an inhibitory nanobody, the effect of an inhibitory nanobody. We measure multiple of these ones. And as you can see here, they do not inhibit. And one very important thing, these experiments that I'm showing here, they are done with 1 nanobody and 1 chip. So in this particular case, we are not using the same chip for multiple ones, just 1 nanobody per chip. An example of an inhibitory nanobody that do not allow transport of choline is depicted here. And these are the ones that you can see here. Like, for example, this one nanobody 5 showed one of the strongest inhibitions of transport. And actually we had some other ones that show even a stronger inhibition. So at this point, actually, we realized that if we were to perform an extensive washing, meaning that after the very last step, we were to repeat the cycle, but without having a nanobody, we actually were able to recover almost recover 100% of the initial signal. And this is depicted here. So the dark blue line here, that's the experiment without nanobody. And then after the application of the nanobody, which is an inhibitory one, you see here the decrease of the peak current. And then after washing, sequential washings of this nanobody, we little by little recover the peak, the peak currents, almost to 100%. Now, this is very important because this was the first indication that one can use this method in a high throughput mode to screen multiple nanobodies per chip. So this is what we did. We performed sequential measurement of inhibition, washing another nanobody, washing again, and another nanobody. And the results are summarized here. So this is now a sequential experiment with different nanobodies using the same chip. And as you can see here, there is an inhibitory one. We can recover the signal, measure another one, another one, another one. So this is actually one of the most important things that to me is really valuable from using this technique for the screening, because these nanobody libraries are usually having like dozens or of nanobodies. So this can become very, very important for screening. Another thing that actually, well, we realize at this point is that if we can use the same chip to perform, to pass different nanobodies, we were also able to pass different concentrations of the same nanobody, meaning that we are able to determine inhibitory constants of this one. So this is the experiment that you can see here. We have, we started with no nanobody, and then we go from the low concentrations so that to not oversaturate the binding site. And then going from low concentrations of nanobody towards high concentrations. And this is, and by plotting the peak currents, you can calculate an IC50 for this particular nanobody of around 18 nanomolar, which is very reasonable, and it agrees with what has been reported for other nanobodies in other systems. So a summary of the highlights of the method. This has the potential to become a high throughput selection method for selection of inhibitory binders, not only for multiple electrogenic transporters, especially those which will be particularly important for those for which there are no labeled substrates available. Now, whether this can be done with monoclonal antibodies or fragments of antibodies, fragments, for example, larger fragments, we don't know. We haven't done this type of experiments, but it could be also, I will foresee that it can be also used for that. Of course, it will be much more cost efficient than screening these 25 nanobodies using the liposomes assay that I described before. And we speculate that we can perform this method also with membrane vesicles. This, of course, will be also beneficial. SSM has been used before to determine currents with membrane vesicles, so you will not need to purify and reconstitute proteins. So that will actually also boost the application on this nanobody selection. And just to not give you a conclusion to this story, I can tell you that we use those nanobodies, many of those, for crystallization again. It turns out that actually they didn't help. They actually, we got worse X-ray diffraction than before, as you can see here. So that was, you know, bad for us, bad for Natalie. It was really, really sad. But later on, we were able to apply single particle cryo-EM now with the nanobodies and determine a high resolution structure of this protein in lipid nanodiscs. You can see here the densities for 2 nanobodies. It turns out that this protein is a dimer in the membrane, and we now can visualize high resolution features. So we went back after that to the X-ray data and to perform molecular replacement using the cryo-EM model. We rephase our X-ray data, and now we have also wonderful density for our X-ray conformation, which is a different one where we can see choline bound in the binding site of this protein. So with this, I conclude this story. Thanks to the members of my group, especially Natalie, which did a wonderful job during her PhD, and Biocentrum, and of course the non-ion people, because we had this wonderful Surfer N1 grant, and the Swiss National Science Foundation, the Helmut Horten Stiftung, which fund my lab, and of course our collaborators, Marcus Ziegler and Cedric Rutter. Thank you very much for your attention.
Thank you so much, Camilo, for walking us through. I now open the floor up for the portion of the Q&A session. If you have any questions, feel free to send them into the chat. So we have a question that's coming in now, just typing away. This question is for Christine.
Sorry, Jason, how can I see the question?
It's just coming into the chat, so I'm going to read the question. So did you consider the effect of salt on the assay? Did you keep the osmolarity constant?
Yeah, yeah, we consider this. The problem is that the whole osmolality question in the SSM is for us at the moment a little bit vague. Yeah, so I have to say that's why we kind of went for this non-physiological stimuli. Yeah, it's a good question. I cannot really answer that. It is something we have now to try also by using now different ways how we apply now the driving force. Yeah, we thought that we are on the safe side by using this ionized sugar. Yeah, but if we can really now avoid osmolality changes, I can't tell. That is an excellent question, which especially for an osmolyte transporter has to be considered.
Christine, can you hear me?
Yeah.
Yeah, hi Christine, it's Björn Petersen.
Oh,
It was my question. I was just wondering, could you use other organic ions to sort of match out the osmolarity so you have the same concentration on both sides? Did you try that?
No, not yet. Yeah, but as I said, that's we are kind of at the beginning with this study because first of all, we wanted to understand what we see, and it took us some time to understand at which parameters we have to look at. But it's definitely on the plan now to go into that. Yeah, but it's yeah, absolutely.
Perfect. Thanks for an interesting talk to both of you, by the way. Very cool.
Thank you, Björn. I now move on to another question for Camilo. And again, I welcome the individual that asked the question. If you want to unmic and ask yourself to keep it more personal, I think that's fine. Totally up to you, but I leave it open. If you would prefer me to ask the question, that's also okay.
Fine to do that, right? I just wanted to ask Camilo whether he validated the inhibition data which you got from your surfer measurements, maybe with traditional liposome assays using radioactivity, just to see whether the numbers are in the same range.
Oh, wow. Now you want us to go back. We haven't done it. We haven't done it, but that's a actually very fair point. I will tell Natalie to go back and do this measurement. Thank you for the suggestion.
Okay, thank you.
And then Camilo, I have another question. Can you observe a difference in the inhibition characteristics of nanobodies compared to small molecule inhibitors? And then secondly, as a follow-up, is the recovery time of the signal different comparing the nanobodies?
So for this transporter, we just have one other small molecule inhibitor, which is a very well known molecule that inhibits choline transporters. It inhibits a human choline transporter, this HC3. And unfortunately, that inhibitor it was not reversibly, it was not possible to get rid of it after washing extensively. We don't know why, so we cannot really compare with inhibitors that are reversible inhibitors. So yeah, and among different nanobodies, we didn't see major differences except for the height of the peak currents.
And as a question for Christine, how can you distinguish between transport and binding in the case of BetP?
Yeah, also very good question. And I have to say, I was a long time not an SSM believer, sorry to say that, because this was exactly my point, how I can do that. And that is exactly why we have these mutant, which I presented here, to see where we can definitely see that there is not transport available, that there's not transport. And at the moment, I would say you will always have a mixture, yeah, as I said, of a pre-steady state and then a real steady state where you can see transport. but as long as we do not have an inhibitor, it will be very difficult really to answer that questions to the very end. Yeah. So we compare it for sure to uptake measurements, and most of them were in fact done by Camilo during his PhD. But it is, at the moment, I cannot tell you this part is exactly now the transport component. Yeah. Let's see where perhaps with different mutants that we can, or with an inhibitor, we can nail it down a little bit better. But it's definitely still something which has to be solved. Yeah. But I'm absolutely convinced we see transport in the wild type, because the transients look very different, and the translocation pattern looks charge translocation looks very different for the mutant I presented.
And then back to Camilo, if the on-off rates for the nanobodies, the choline transporter, are relatively low, could you use the SURFER instrument to measure or confirm these rates? And if so, how do they compare to the SPR data?
Yeah. I guess that can be done. Yeah, we haven't analyzed the data in looking at that, but I will say that's totally feasible. So we will need to do it, because we have now both data, also the SPR data. So yeah, we will give it a try. We haven't done it yet.
And then for Christine, regarding the sensors, would the higher time resolution of 1 millimeter sensors help distinguish binding and transport better? Did you try this?
Yes, we tried it, but unfortunately, this mutant was so fast that even with a 1 millimeter sensor, it was not possible. But it is definitely the first thing to check, to go from a 3 millimeter to 1 millimeter. You saw differences, but it's still, we even with that sensor, we were at the resolution limit.
And then back for Camilo, in the case of unspecific nanobody binding, how can this be prevented during the SSM experiment?
Thanks, Jason. That's a very nice question. So we will recommend, and actually this was suggested originally by Randy Stockbridge, which was here talking in the last seminar. She recommended to use a BSA in your buffers, to include a BSA before applying a nanobody, so that this can bind to unspecific sites. That I agree with her. Actually, this can be a good way to deal with this. Yeah.
Okay, I know that we're a little bit above the scheduled time, but I leave the Q&A open for any other last questions. Another one for Camilo. Did all the nanobodies you screened bind to this same epitope? Can you choose the epitope?
We have determined cryo-EM structures with 2 different nanobodies. Those 2 bind to different epitopes. I cannot tell about the rest, and I don't think we will attempt to solve 25 cryo-EM structures. So at least those 2 bind to different epitopes. Yeah.
I'll leave the Q&A open for some last questions. Again, all of this will be transcribed and be made available on demand. And as such, you can always get in contact with us and the presenters if there's any questions that come up after you rewatch today's session. So I just leave the Q&A open for another minute. And then as a reminder, also, because it was touched upon, session 1, session 3 now will be on the 1st of July, that's Thursday. And registration will be open after today's event. So everyone that registered for today's event would get a notification of the on-demand copy, as well as notice of the upcoming event in July. So with that, I would close today's session. Again, from my side, and I personally want to thank the presenters for their contributions, both great presentations, and we look forward to any additional questions that is addressed to them at a later state. If there's any other questions regarding anything that we've gone through, whether it's the process of the ongoing SURFR N1 grant campaign, please don't hesitate to get in contact with us, and we wish you a good rest of your day, wherever that may be.
Thanks, Jason.
Thank you.
Thanks. Bye. Take care. Bye-bye.
Bye.